There we go. Thank you all for attending. Excuse me, a few acknowledgments right off the bat, and I do apologize in advance. Unfortunately, I'm fighting a little flu or cold or something, so I'm going to sound a little raspy through this. But I am David Moss with the Nature Conservancy, and I certainly want to acknowledge our partners in this project, the South Atlantic Fishery Management Council, LGL Associates, excuse me, South Carolina Department of Natural Resources, and Dr. Will Heyman, and also certainly our angling partners in each area that without them we couldn't have accomplished this. So thank you all, and let's get right into it. Excuse me, next slide. Spawning 101. Most fish reproduce through a process called spawning, which is where the female fish will release her eggs into the water. The male fish will then come in, of course, and fertilize the water with releasing their sperm into the area. A lot of fish tend to form together in tightly packed schools. We call these spawning aggregations. When I say fish, a lot of the snapper grouper species. So we're looking at some areas that are known spawning habitat for some of these collections. Next slide. As we said, we're looking at some specific areas. A lot of these fish, when they do aggregate, it'll be very specific geographic features that we call elbows. And anglers, of course, were experts at finding where some of these areas are. Combination of some of these known spawning areas, along with the fact that these fish do aggregate, can certainly make some of these fish vulnerable to overfishing at times. As we as fishermen know when this is going to occur, usually it's around moon phases and whatnot. So it's very easy for us to go to these areas and really concentrate our efforts in an area where we know that there's going to be tightly packed schools of fish. Excuse me, next slide. So what can we do about this? In 2010, the South Atlantic Council began discussing spawning special management zones as part of what we call EBFM, or ecosystem-based fisheries management, or multi-species management strategies. Through the help of anglers, we identified a couple of what we call SSMZ sites, spawning special management zones, and we looked at times and locations for some of the key species, again, through angler partnerships with this. Some of the things that we also looked at were ocean depth and underwater landforms, as we discussed just previous, and then also looking at some independent fisheries data as well. Next slide. So these SSMZs, 5 sites were established in 2017 through Amendment 36 to the Snapper Grouper FMP. The goal is to protect fish spawning in these areas. The protection sunset in 2027 for 3 of the natural reef sites, unless we can provide evidence that these sites are effectively used by these spawning aggregations. And as you can see in this graphic here, the 3 natural bottom sites that we looked at were South Cape Lookout, just off the coast of North Carolina, Devil's Hole, just off of South Carolina, and then Warsaw Hole, which is south by southwest of Key West down in Florida. Next slide. And as we stated, the clock is ticking. System Management Plan Work Group anticipated receiving this data from existing sampling programs, and the check-ins were scheduled for 3, 5, and 8 years, but unfortunately only limited monitoring has occurred. Existing sampling programs wouldn't have provided the data that the council really needs to help make their decision for 2027. So that's where we, again, had partnered with the council to look at some of these areas and determine whether or not they were effective spawning grounds. Next slide. Some of our objectives: determine seasonal sampling and whether these priority species were abundant and spawning, learn from local anglers where they're finding these fish and at what times of year, and then of course share the results with the South Atlantic Council and the public to help inform the decision making on the sunsetting protections. So some of these target species that we certainly wanted to look at were red grouper, scamp, gag grouper, Warsaw grouper, and greater amberjack. Oftentimes these sites will have multiple species aggregating throughout the year. So while these are kind of the highlight species, we were certainly open to seeing what else was around and if there was spawning activity occurring. Next slide. Next slide. our timeline. So in 2023, as you can see, we did one sample of Georgetown Hole, and that was our first. And then in 2024, we were able to do our second sampling at Georgetown Hole, and then our first at Warsaw Hole. And then in 2025, we were able to do 3 trips, one more in Warsaw Hole, and both of the trips to South Cape Lookout. And you can see, excuse me, in the graphic below, the funding for each of these trips and where that came from. And next slide. With that, I'm going to turn a lot of the more technical aspects of this over to Lewis. So Lewis.
Alrighty, everybody hear me okay?
Yes.
Oh, perfect, thanks Chip. Yeah, and thanks David. So our group at LGL Ecological Research Associates was contracted to conduct the field sampling. And on behalf of the wider team and my co-investigator, Dr. Peter Mudrak, I'm pleased to report on the methodologies that we utilized and of course later the results of those efforts. Next slide. So in brief, we used standard cooperative monitoring techniques for sampling spawning areas in the US South Atlantic. This was a protocol that was developed by Dr. Will Heyman back in 2016. Now, the cornerstone to this approach is partnering with local commercial and recreational fishers who know these areas better than anyone, and many of which were strong advocates for the implementation of these SSMZs to protect spawning fish in the first place. Now, these included Captain Jack Cox out of Morehead City, North Carolina, Chris Conklin and Captain Andy McGraw out of Murrells Inlet, South Carolina, and Brad Nowicki of Off the Rock Charters in the Florida Keys. Next slide. So we used 2 primary techniques to sample within the reserves. These were first kind of underwater video surveys to look for spawning activity, for fish aggregations, as well as for indicators such as kind of courtship behavior and courtship coloration that can be indicators of fish spawning. We also used conventional hook and line sampling to obtain reproductive samples or gonads, to extract the ear bones or the otoliths for aging, and to collect morphometric data, so fish lengths and fish weights. In addition to this, we deployed multi-parameter sondes to characterize the environmental conditions at the time of sampling in terms of water temperature, salinity, and oxygen levels. Next slide. Now, as I'm sure many of you are aware, in most temperate environments, spawning is largely seasonal, kind of comprising of peaks in activity during certain times of year, followed by inactivity or regression. Now, these peak spawning windows can be fairly short, with some reef fish species also exhibiting lunar-related spawning that coincides with certain moon phases. So that's a lot to take in this table here from Farmer et al. 2017. But really the key take home is that the months that are shaded in black are months when predicted peak spawning activity is occurring for each of these respective species on each row. As you can see, peak spawning often comprises of just a few months for some of these species. Now, as an example, in the second row there you have greater amberjack, one of our priority species at some of these sites. Now, spawning in this case is predicted to peak in April or May, and this often occurs alongside the full moon. Now, sampling within these SMZs, therefore required a kind of targeted approach that was really scheduled to coincide our sampling within these predicted spawning windows and times for the species that we were interested in. So to do this, we used a suite of information, including published literature, fishery independent surveys, and local fisher knowledge, particularly from that of our kind of cooperative research partners, to really hone in our sampling trips and plan them accordingly. Next slide. Once on the site, we tried to pinpoint additionally areas within each reserve, particular sites that we were interested in visiting. Now, to do this, we use local bathymetric mapping to identify features such as ledges, and promontories that might hold fish. We incorporated historic sampling data from within the SMZs that was collected by both LGL and the Council prior to 2017 and in 2018 in the case of Warsaw Hole. And we also used real-time sonar detections while within the reserves to identify suspected aggregations or activity hotspots as interpreted by the captains. Next slide. So this is just a brief overview of the 2 sampling trips that we conducted at each of the 3 sites. Each table denotes the month and the year that sampling was conducted and the chosen priority species. So as a reminder, that was scamp and gag in Georgetown Hole, Warsaw grouper and AJs at Warsaw Hole, and red grouper and AJs at South Cape Lookout. And the data points on each of the 3 maps further show the areas or the sites within the reserves that were sampled. And as you can see, many of these were concentrated near various bathymetric features. And at South Cape Lookout, I'll also point out, and that's the map on the far right, I'll just point out that we also opportunistically sampled some sites adjacent to the SMZ boundary. Next slide. So to determine whether these protected areas are serving their intended purpose, our mandate was to obtain evidence of spawning from within the reserves. And for simplicity's sake, that this can kind of be broken down into indirect and direct evidence for spawning. So indirect evidence constitutes things like courtship coloration, courtship behaviors that we observe on our camera surveys, things like this gray-headed behavior in the scamp pictured on the image to the far right, that is thought to be related to courtship behavior for that species. The other lines of indirect evidence also include higher than normal numbers of individuals of a species within the area. Next slide. So direct evidence includes video documentation of fish actively releasing gametes into the water column. Right, and that's really the holy grail of spawning evidence, where timing for everything has to align perfectly. Now, fortunately, direct evidence can also be obtained by collecting reproductive tissues and inspecting those under a scope. Now, which really is kind of the perfect time for me to hand the baton here over to Kevin from South Carolina DNR, who led those analyses. So, thanks.
Yeah, thanks for that, Lewis. You guys hear me okay? I'm going to turn my camera on here too.
Yep, we can hear you.
All right, great. Yep, thanks again, everyone. My name is Kevin Kolmos, and I specialize in fish reproductive biology here at SCDNR. We were happy to be a part of this project. Our program was specifically contracted out to process the reproductive tissue and then interpret those reproductive phases of those individuals. So I'm going to go ahead and give some background info on how we accomplish those tasks. Next slide. So first, let's discuss the fish reproductive cycle. During a fish's lifetime, a juvenile fish will mature once and then participate in either one or many reproductive cycles. Most of the offshore species in our waters are considered multiple batch spawners, and that means that they can spawn multiple times, not just over their lifetime, but also within a spawning season. And a spawning season, like Lewis had mentioned, can last months. And we can tell they do this by collecting many fish samples. And specifically, we're looking at the reproductive organs called the gonads, and we look for what is called spawning indicators. And I'm going to explain that a little bit more in some of the upcoming slides. But for now, let's zoom into one of these reproductive cycles, and I'll explain the different phases of reproductive development. And next slide, thanks. So this is a visual for the fish reproductive development where gametes must develop through maturation for spawning to occur. And by gametes, the female gametes are the eggs, which we call oocytes, and male gametes are spermatozoa. So this visual is from Lowry Barbieri et al. I was a co-author on this paper, but guides like this help us to categorize and standardize the phases of reproductive development individual fish are in. So let's go through this guide. When a fish first enters the mature population and participates in the spawning season, first we're going to see the gonad in this early developmental phase. And this is where the fish is receiving social, behavioral, hormonal cues to start preparing that gonad for spawning. So for females, they're going to continue to develop those oocytes through the late developmental phase, and then hopefully those oocytes will mature and spawn. And for males, spawning is going to occur when they develop their gametes through the process of spermatogenesis. And that's when they release their spermatozoa. After spawning is complete, the fish enter the regressing phase, and that's kind of just the shutting down or the end of the spawning season. And then lastly, the individuals will enter the regenerating phase, and that's where they're just kind of doing some cleanup and they are preparing the gonad for the next reproductive cycle. And there's little to no development in that phase. But before we can even categorize the gonad tissue, we have to process and interpret the tissue through a process called histology. You can go to the next slide. Histology is simply the study of microscopic tissue, and in gonad histology, we're microscopically looking at gonad development to determine what reproductive phase each individual fish is in. So for this study, LGL collected those samples from the fish and fixed the gonad tissue before sending them to our lab. Fixing the tissue is just a term we use, which means we're preserving that tissue and stopping the tissue from degrading. And in our case, we're using formalin for that. Then we go through several processing steps where we embed the gonad tissue in paraffin wax. That allows us to take extremely thin sections of the gonad before placing those sections on a microscope slide. Then we're going to stain the tissue and bring out the many characteristics of the gonad, and that's going to allow us to better interpret the slides. So we have two readers that stage each of our sample and come to a consensus on the reads. And that whole process takes about 2 months. Next slide. So when we're analyzing our histology slides, we're focusing on many characteristics of the gonad because we want to correctly assign reproductive phase. And this step underlies our ability to estimate parameters such as maturity and spawning seasonality. We base our staging partly on the most advanced gamete within the sample. So in males, we're looking at the presence and degree of spermatogenesis, and in females, the oocytes that are farthest along in development. So for this study, we're particularly interested in the spawning individuals. And in spawning males, they will contain spermatozoa. And for females, we're looking for the spawning markers, which include oocyte maturation, hydration, and the presence of something called post-ovulatory follicles, or POFs. And the POFs are left over from released eggs, which means that a fish has recently spawned. Female spawning markers in many of the species we're studying generally only last about 36 to 48 hours, so the timing of capture is very important. For most studies, we're focusing on the females in analyses. Well, to be honest, we all know that females are more important anyway, but we do focus on egg development because females are usually the main drivers of reproductive potential or the number of offspring that can be produced. So next slide. All right, so I gave you a crash course in the methods and interpretation, so everybody online should be experts at this point, but now, of course, there's so much more to these processes, and there's a lot of great resources and visuals which provide detailed information on terminology, the characteristics of the gonads, as well as a framework to evaluate fish reproduction. So for example, these visuals show the progression of gamete development in both males and females. And this is part of that same paper that I showed earlier, and there's a QR code there. Hopefully that works if you want to take a deeper dive into gonad histology, since this information is pretty critical in assessing fish reproduction. Next slide. So I'd like to give you some very basic results by taking the visual guide I showed at the beginning of the presentation, and then we separated individuals into these phases based on those most advanced gametes and other gonadal characteristics. Go on the next slide. Oh, the animation is going around. Yeah, there you go. All right, so this pie chart represents all the females caught during this process. And I pulled out the immature fish so that you can see the resulting sample size, which is just below each of those reproductive phases, along with the percentage of fish in each one of these categories. So this is just kind of a teaser, and I'm gonna pass it back over to Lewis, who's gonna give you a deeper dive into what these results actually mean at each one of those sites that he discussed.
Yeah, thanks, Kevin. So I'll go into the kind of indirect and direct evidence for each of these 3 sites. So next slide, please. The moment you've all been waiting for, of course. Okay, so starting with Georgetown Hole. Georgetown Hole is a special site with areas of kind of complex high relief bottom habitat and these rocky outcrops that really do harbor a diverse reef fish assemblage. So in April 2023, we returned with 55 samples, including some of the species listed, such as speckled hind, gag, scamp, snowy grouper, among others. And then, following that in April 2024, we again returned with 89 samples that covered, again, much of the same reef fish assemblage, but also included the likes of Warsaw grouper. Next slide. In terms of the indirect evidence at Georgetown Hole, camera surveys captured scamp exhibiting kind of courtship coloration and courting behavior at 8 sites across the reserve, with individuals like this one pictured here in the bottom left, expressing that kind of typical gray head and nuzzling a larger, darker individual that's obscured in the background of that image. We also observed the presence of large black belly gag, as pictured on the right within the reserve, which is a nice indicator of a kind of healthy, mature adult population at the site. Next slide. So the biological sampling, our direct evidence for, we basically got a good chunk of direct evidence for spawning in April. For instance, 100% of the scamp that we sampled in 2023, so 7 females were all in spawning condition. And again, 93% in 2024, which was 14 females. I think is also particularly encouraging is that the age of the scamp that we sampled was relatively large, a mean age of kind of 9.5 and 10.5 years old, with some individuals being as old as 15. We were also we were unsuccessful in collecting any gag in 2023, but we did manage to collect 7 females in April 2024. 8 females in total, 88% of which were in spawning condition, for a mean age of 8.9 for gag grouper. We were also collecting some blackfin snapper that showed direct evidence of spawning. So 50% of the blackfin snapper that we collected in April were in spawning condition, 33% during our 2024 sampling. Next slide. And so that's just kind of a quick high-level summary for Georgetown Hole here. So scamp, gag, and blackfin snapper are all spawning within the reserve. Warsaw grouper and speckled hind are present within the reserve. I think this is an important finding, given that one of the additional goals of the SMZs, as laid out in Amendment 36, was to reduce bycatch and bycatch mortality of these species in particular. Large male gag were also present within the SMZ, along with advanced age fish of several species. Now, gag are protogynous hermaphrodites, beginning life as a female, and then transitioning to male later in life. So, I really can't kind of understate that the importance of this finding that we were able to collect gag, and given our relatively small sample size, especially given that the last stock assessment for the South Atlantic estimated that male gag accounted for approximately 5% of the population. Next slide. So on to Warsaw Hole. It's really another unique site that certainly lives up to its name of being an important area for Warsaw grouper. It's pictured on the right, but also for amberjack, as you'll see shortly. So in June 2024, we returned with a total of 35 samples, including some of those priority species such as Warsaw grouper and speckled hind and scamp and these other species listed here on the slide. Now in April 2025, fishing conditions were pretty tough. The currents were relatively strong and the sharks were pretty relentless at depredating many of the fish before we got them to the side of the vessel. But we did return with 20 samples and we were successful, including our priority species such as Warsaw grouper and greater amberjack within that sampling trip. Next slide. So our April 2025 sampling at Warsaw Hole was targeted the full moon, and really we were most interested in greater amberjack. So one of the things we did in terms of collecting indirect evidence was drift our camera system through suspected amberjack aggregations that we identified on the sonar, as pictured on the right here. So we could actually observe our camera system on the sonar and kind of adaptively change the depth so that we were drifting the right depth profile to hopefully target and capture some of the imagery of some of these aggregations. We did this oftentimes in the evening when the fish seemed to be more active and up in the water column. And this approach was highly productive, and we were able to collect some great footage of amberjack in high densities within the reserve. So next slide. This video, which I'm hoping perfect, started playing. So this kind of illustrates one of these encounters. You'll see these little white specks in the distance that will slowly come in closer and closer. These are the amberjack, highly curious critters. In this particular camera drop, we estimated a total of, I think, 47 amberjack in a single video frame. And we observed aggregations of AJs like this across multiple sites within the reserve during our April full moon surveys, which is really encouraging to see. I'll let this one play out here a second. Of course, we're extremely lucky with the conditions for filming, with the water being nice and clear. Okay, so next slide. So that's the indirect evidence. How does that pair with our histology sampling? So going back to June 2024, we collected red snapper that were in spawning condition, but the two Warsaw grouper that we sampled were regressing and developing, so they were not actively spawning at that time. Interestingly, though, during our June trip, fishers in the Keys did report to us that they kind of incidentally catch Warsaw grouper high up in the water column while fishing for amberjack in April. And this is a behavior that we thought could be related to spawning and egg release. So in line with that, those anecdotal accounts, during our April 2025 sampling, where again we were targeting greater amberjack, we did catch two female Warsaw grouper within the reserve. And this time, each of those individuals was determined to be in spawning condition. So this provided direct evidence for spawning of Warsaw grouper within the reserve, but also implied that peak spawning, that window of peak spawning at least, may start slightly earlier than previously anticipated for this species. And lastly, as expected from our video surveys with the high numbers of greater amberjack at the site, many of the females were also rode up and in spawning condition in April, which really nicely kind of pairs with the video footage that we collected. So next slide. So quick high-level summary for Warsaw Hole. Warsaw grouper are spawning in the SMZ in April, as are greater amberjack, which are aggregating in relatively high densities and spawning. We also collected advanced age red snapper, with some individuals being 19 and 20 years old, 21 years old, that we collected within the reserve. And then in addition to that, both speckled hind and black grouper were present within the reserve. So next slide. So moving on to the last of the three sites, South Cape Lookout. Our South Cape Lookout sampling trips with jack were highly productive. I think that's one of the many perks to working with commercial fishing partners. In our April 2025, we returned with 72 samples, including species of interest such as red grouper and scamp and gag and greater amberjack, as well as the other species listed on the slide. And in May, again, we outdid ourselves and returned with 100 samples, comprising many of the same species, and again, including red grouper, which of course was a species we were particularly interested in at that site. So, next slide. So in contrast to the other two natural bottom SMZs that I covered, I do want to point out that South Cape Lookout had far fewer areas of high relief within the reserve, meaning that fish tended to be concentrated in a few select areas. Other areas of the SMZ is kind of pictured in some of those images on the right, were instead kind of characterized by a lower relief habitat with interspersed sandy bottom. Now, these conditions were considered possible red grouper spawning habitat when the SMZ was established, given that red grouper are thought to excavate spawning burrows in these softer sediments. Now, with respect to indirect evidence, for spawning, we did observe high densities of vermilion snapper within the reserve during our May camera surveys, and I'll talk a little bit about that shortly. Next slide. So our sample size for red grouper were generally low, about comprising of 2 individuals, I think, during each trip. But both the male and the female red grouper that we sampled in April 2025 were actively spawning. And what was particularly impressive to us was that these individuals were 19 and then 22 years old, which really is a long time for a fish to avoid a hook. In April, a small number of female gag, silk snapper, scamp, and blackfin snapper were also determined to be spawning within the reserve. And in line with those kind of aggregations of vermilion snapper that I mentioned us seeing on our video surveys in May, Histology also revealed that a large number of female vermilion were spawning, total of 18. During our May sampling trips. And in addition to that, we also collected samples that showed spawning in almaco jacks, blackfin snapper, and scamp. Next slide. So as a quick summary for South Cape Lookout, so large red grouper spawning in the SMZ in April, although apparently at low densities. Scamp, gag, blackfin snapper, silk snapper, vermilion, and almaco jacks are also spawning within the SSMZ, and we collected evidence to show that there are many advanced age grouper, with red grouper at 23 years old, scamp of 21 years old, red porgies are coming in at 12 years old in some cases, and even gray triggers as old as 10 years. I was also personally particularly impressed that we collected, a 13-inch Graysby that was aged at 19 and a 16-inch Rockheim that came in at 21 years old. So really remarkable that the age of some of these samples that we collected. And lastly, I just want to point out too, is that particularly in South Cape Lookout, we did collect a decent number of immature snowy grouper that appeared to be present within the reserve during both of our April and May sampling trips, which may indicate the area potentially being an important growth habitat for that species too. I think with that, I'm handing it over to David just to kind of tie a bow on this and provide some high-level conclusions.
Thank you very much, Lewis and Kevin. So just as a very brief overview of all the information that we just took in, we did see demonstrated spawning in all 3 of the natural bottom SSMZs that we sampled. So we are protecting sexually reproductive fish in these areas. Excuse me. And of course, multiple species use these areas that we've seen, as evidenced by all of the findings that Lewis just went over. So there's certainly strong evidence that these areas are effective spawning grounds, and that these, even though they're small, that they are very effective areas as we looked at. And with that, I will open it up for any questions if anybody has for the group.
Before we do that, let me go ahead and go over how to operate the webinar again, just to make sure everybody has an opportunity. If you're using the web app, click on the hand icon at the bottom left if you'd like to ask a question. In order to raise your hand, just click on it. It should turn white. If you're using the desktop app, if your hand is red, that means your hand is up. And then we will recognize you to ask the question. As the process goes along, the webinar will notify you that you're next up to speak. Go ahead and click on your microphone. If it has a slash in it, that means we're not going to be able to hear you. However, if it doesn't have a slash in it or it's green, that indicates that we will be able to hear you. Or you can type questions into the question box. You can find it here, which is at the top right, and then there'll be a box down at the bottom. So with that, let me go through this. I will open it up to any questions that the group might have. But let me start off by saying thank you very much for the team presenting here today. I think it was a great overview of spawning and spawning special management zones. Gave a lot of information, and it's good to see that data has been collected over the past few years. All right, not seeing any questions, so I will ask a question, likely to Kevin. When you're seeing indications of spawning with these fish in ripe stages, I think you had said that spawning was likely to occur within 48 hours, is that correct?
Yes, that's correct. So when we're looking at oocyte maturation and the presence of POFs, that entire process of developing those oocytes in kind of the final stages before being released, and then the time it takes for those POFs to kind of resorb or degrade within the gonad, that whole process is around 36 to 48 hours, depending on the species.
So just curious on how you all came to know like the time period for these spawning events and the time series for figuring out when spawning occurs.
I think that is mostly done with specimens in captivity. So they can time spawning, they know when eggs are released, and then they sample fish at certain time periods after the known spawn time in order to see histologically how much those POFs degrade. And then also you take samples before spawning occurs, and again, kind of timing how long those different processes are. I mean, actually developing the oocytes from the time of early development until they're kind of reaching that oocyte maturation stage, that can actually take weeks, if not longer. So I think there are a lot of other social and behavioral cues that the different species are getting when they're out in the wild. But as far as what we can tell from some of these captivity studies, as well as just the numerous samples that our project has collected, that's kind of how we determine the I guess the ages or the time frame of those different reproductive phases.
Thank you. See, Wilson has a question or a comment. Go ahead, Wilson. Wilson, it looks like you have two different webinars open. So if you turn off the sound on one, let's see if this works.
Yeah, let me.
So Wilson, the one with your hand raised, I'm going to keep you on that, and I will dismiss you. Well, you're not making noise now, so go ahead.
Can you still hear me?
So Wilson, I'm going to dismiss the other Wilson that's on there because that seems to be making the feedback. And And let's see how that works. If I accidentally kick you totally off, please rejoin. All right, how about now?
Can
Go
you
ahead,
hear me?
Wilson. Yes, we can.
Oh, good. Okay. So I can't see you anymore, but that's okay, as long as you can hear me. So my question was, I know your sample sizes were pretty small, but did you get a pretty good range of ages of spawning females on the sites?
Yeah, I can answer that one. So I think it's important to acknowledge our sample size, especially for some of these species like Warsaw grouper, right, where our samples are relatively small. So to answer your question, the answer is yes. For some species, such as scamp in Georgetown Hole, I think we had sufficient sample sizes to look at, age frequency of females. But for the most part, I'd be cautious on, drawing any real conclusions on ages for a lot of these species, just due to the limited sample sizes. I think, with the caveat being, well, the exception being scamp, Georgetown, vermilion snapper at some of the sites, and some of the other snapper species, the blackfin snapper, that we seem to encounter a lot of.
Thank you.
You're welcome.
What were the ages?
For which species in particular, I guess?
Well, for scamp, I guess, since you seem to have a better sample size for that one.
So for scamp, I covered the mean ages, which I think was around, let's see, it was slide 8. It was about 8 or 9 years was the mean age for scamp. Our sample size was about 20 individuals, and I think the oldest one was 17 years old.
Oh, okay. And do we presume that, as is the case with so many species, the older, larger females are the most desirable ones from a spawning standpoint, that have produce more eggs and maybe more viable eggs?
That is a great question. Regarding viability of eggs, I'm inclined to let kind of Kevin chip in on that, given that they conduct the histology. Kevin, do you see any patterns regarding age and viability?
Yes, and I would kind of relate that more to a size metric, whether it be length or weight. We definitely see increases in that reproductive potential based off of the size of the fish. Obviously, if the fish is larger, its body cavity can be larger, its gonads can be larger, and it can release more eggs and produce more eggs. There's been a lot of studies done on a range of different species that show that larger individuals actually start spawning earlier, can spawn later into the season, and can actually spawn more frequently than maybe some of the smaller individuals within the population. Again, it's species specific. There can also be just fluctuations in spawning frequency from year to year. Sometimes there's enough food, the conditions are ripe for growth and energetics. So there's a lot of different I think inputs that go into the success of spawning, and we actually see that where there'll be some banner years of lots of baby gag grouper around, or just lots of baby red snapper around, and we're not always sure why the conditions were right for those recruits, those eggs to be so successful. But we do see those changes, and we can for sure say that large individuals are most often able to produce more eggs.
So one more follow-up. So from a management perspective then, should we, when I say we, the fisheries management family, be trying to target for a certain percentage of larger, older females in a given population? And do we have any idea, what that percentage should be in order to enhance the sustainability of a given stock.
When you say target, do you mean protect?
Well, no, I was thinking more of the age structure of a given stock. So should we, for example, say that we need at least 10% of the gag stock at ages of 15 or above?
Gotcha.
Is it possible to actually come up with some management recommendations that would enhance sustainability by ensuring that you have a certain percentage in those older age classes?
Yep, no, that's a great question, and those are the types of questions that go into the stock assessments that we have, through the Management Council, and those are the really important inputs that will help us make sure that we're having that renewable resource. So I would say, yeah, you definitely are going to want to protect a certain portion of the population, and the reproductive biology, along with the age and length information that we collect, are all absolutely crucial in order to provide inputs for the numerous models that go into these fisheries assessments. So, I can't give you definitive percentages or numbers because they're going to vary based off of the different species. But that's that's definitely a factor that goes into these assessments.
Okay, thank you.
Yeah, and Wilson, we do have MSY proxies, and those are based on spawning potential ratios. And so those are included in stock assessments when maximum sustainable yield cannot be estimated through the stock assessment. So there are targets that are developed, and those are incorporated into management. And the next question is from Laurent.
Hi everyone. Yes, so so those spawning aggregation sites usually they're related to a certain population or subpopulation, and so I wanted to ask if you guys have an idea of what sort of population of where the species are distributed that those aggregation represent, because it may be specific to the area, but how is the population outside of that spawning population? My question is how it relates to the rest of the population, and how do we get an idea of what's the status of the spawning stock, for instance, is in between those two sites, and where are the fish coming from that goes to those aggregations?
Yeah, great question, and we would love to know the answer to some of those questions too. But it's important to think about it, and so we're kind of confined by funding for these sampling trips, and so one of the things you'll note from the presentation is that for the most part, our sampling was within the box of the reserve for those SMZs, and that was in part the context that David provided during his introduction was that very few studies had looked at spawning within those reserves since their implementation in 2017, and so we very much focused our sampling within the reserves, and we didn't really enact some sort of backy design where we were looking at how the sites or adjacent sites as kind of controls to look and see if the reserves had higher numbers of various reef fish species than areas outside. What I will say is that there was some sporadic sampling that occurred from kind of 2014 to 2016 during the times when the reserves were implemented to kind of inform the exact placement of the boundaries for those 3 sites. And so there's a little bit of prior information, but it's not all that extensive, and it doesn't just cover the areas that ended up becoming the reserves. It also covers some of the adjacent areas too. And then I guess your question was 2-part, and you talk about movement, and so yeah, we haven't done any telemetry studies, be it acoustic or satellite telemetry, to kind of look at residency of these individuals in the reserves or movement between areas nearby or other locations. I think what we don't know if these individuals are there a year-round residents or if they're transients that come in for just a short period of time for spawning. And that's a really important question. I think within this kind of paradigm of these SMZs is if the goal is to protect some of these older, larger, mature individuals, if they stay in the reserve year round, then that's a much simpler thing to do than if they, move and migrate to adjacent areas. Great question. And sorry, I couldn't give you a good answer in terms of data prior to the 2017.
No worries, I appreciate your answer. Thank you.
And Brendan Rundy had a little bit follow up to that as well. Brendan?
Hey, thanks, Kip, and thanks, Laurent, for the question. This is Brendan Rundy with the Nature Conservancy, a co-investigator of this study. And I just wanted to clarify that our objective in this overall study was not to determine whether these areas are the best areas to protect out of all the areas on the continental shelf. It was to evaluate whether these areas the areas that the council designated are at least protecting what we had hoped they would protect, which is spawning individuals of certain species. And just to reinforce what's already been shared on this webinar, we did find that for all three areas investigated, there were members of target species in spawning condition. The finding of old individuals for certain species is icing on the cake, but just to say that, again, these are not necessarily the greatest areas on the shelf. We don't know because we haven't looked at every nook and cranny of the continental shelf edge, but we did find that these areas do have what we hoped they would have and what we hoped they would protect. And so it's up to the council whether they're merit continued protection after the sunset. Thank you.
All right, I'm going to ask a question. I'm not seeing anything in the question log or anybody has their hands up. So if you have a question, please either enter them into the chat or a question box, or raise your hand and we'll get to you. Louis, I know part of the sampling was being done with cameras. In some of these areas, it's quite deep. Were you having any issues with the camera trying to get good images at depth, or were things working out fairly well for you?
Yeah, great question, Chip. It's a tough one, right? Because some of these sites, at least the deeper edges of these sites, you get to a point where light starts to diminish. But actually adding artificial light to better observe what's going on can, inherently affect behaviors and potentially detract species from your camera system. But what I would say is for the most part, the conditions were sufficient for collecting imagery that we were able to use to identify down to species. There were one or two exceptions, I think, when one of those being a trip to Georgetown Hole, when currents were just ripping and the turbidity level was so high that just the camera surveys were ineffective. And so for that one particular trip, which is the April 2024 trip, we largely, also I think it was around the time of the eclipse coincidentally, but anyhow, we largely focused on hook and line sampling because our camera surveys were ineffective. But for the other 5 sampling trips to those 3 sites, we were successful, I think, in getting reasonable imagery to look for indirect evidence of spawning.
All right, Tom has his hand up. Tom, you're unmuted if you want to unmute yourself. Click on what looks like a microphone. It should unmute you.
There we go. Can you hear me?
Yes, we can.
Okay. I have kind of 2 questions in one. Can these sites that have been set aside to be sampled, can they lose their attraction for fish because of things like sand moving into them and covering up, live bottom or whatever's in that area that attracts the fish? And if that is the case, wouldn't that make it pretty important to be looking for other spots to have as test sites as well, sort of developing a larger stable of sites?
Yeah, it's a good point. I think what that raises is the importance of longer term monitoring and regular sampling, so that you can kind of have data points almost annually to evaluate those sites and how they might change through time. I do think that for these deep water sites, they're not as dynamic as perhaps some of the more inshore sites in terms of movement of habitat and sands. And so my general sense would be that there's a reduced risk of these relatively high relief rocky outcrops kind of disappearing or being inundated with sand, at least on the on a reasonable timeline, in which we're kind of interested in here. But that might not be the same case for some of the more inshore sites. I'm not sure.
And just to chime in on some of those other SMZs that weren't looked at particularly in this study, I actually did my master's thesis on Area 53, which is composed of concrete reef cones. And over the few years that I worked on that site, there was definite movement of sand where some of those reef cones were actually being kind of covered and uncovered over time. And I think that is just the kind of natural way of things, and that if you don't have a lot of higher relief, you might have some of that hard bottom covered up. So that is another really good question that kind of goes into this whole study, and really just the effectiveness of different sites.
One other footnote sort of that I'll add to that is that all of these sites, excuse me, when they were initially brought up, were essentially brought to the purview, if you will, by anglers. So as you mentioned, looking for other sites that maybe have these sort of prominent features. Yes, I guess is the easy answer, but it's going to be incumbent upon the angling community, commercial, recreational, and the like, to identify these sites and decide ultimately if they're in need of some sort of protection or something like that. But again, all these sites were brought to the council from fishers.
Thank you for the question, Tom. Did that answer your question?
Yes, thank you all.
Excellent. Anyone else have a question? Go ahead, Laurent.
Yes, so I mean, through your surveys and using the cameras, did you get a count on those different species?
Yeah, thanks for the question. So yeah, for all of our surveys, we have our fishery scientists review the footage and produce counts, either max N or mean N, for basically anything we're able to identify on those cameras. That's something that was not included in this presentation, but yes, that is data that we collect during these surveys.
And then just to follow up on that, the surveys lasted a few days, or just one day, or several days, or I'm asking if you see some variations. Let's say, if you follow the movement of fish from the aggregation in and out of the aggregation over let's say the spawning week, there may be some variations in terms of numbers, and also there are some diurnal variations where the fish are not necessarily there all the time except for spawning. Is there any indication of such things, or how long did you measure for?
Yeah, great question. So generally, a trip and survey would be 2 or 3 days. In the case of Georgetown Hole and South Cape Lookout, where we utilize commercial fishing partners, those are often overnight trips. So we would transit out, we would sample for a full day, and then we'd sample the following day and maybe a half day before transiting back. And the site down in the Florida Keys, Warsaw Hole, where we used a charter captain, we would go out for single day trips, and normally that was 2 or 3 days in a row. In terms of, anecdotally, or did we notice differences in what we were seeing on the surveys? It's a pretty small temporal window, so nothing really striking stuck out. With respect to aggregations, that the one instance I would say that truly meets that definition, I think, was some of that footage that I showed you from Warsaw Hole, right, where we very clearly saw large numbers and aggregations of greater amberjack at the site. And that was something that we saw, at least on the sonar and in part on our camera surveys during, I think, most of the days that we sampled that site in April. So there was no obvious, the fish were there one day and they weren't the next. But again, I think that's as you would expect, given that our sampling really only just covered a couple of days within a given month.
Okay, and then finally, last question is, what do you think, in your opinion, and as to all three of you guys, would be the best method to sort of monitor those, incoming movement and then presence, absence of those fish, and also, you may want to be able to use acoustics, for instance, but outside of acoustics, what would you do?
Yeah, that's a great question, and you kind of took my answer. So where my brain initially would go would be passive acoustic monitoring at the site. Obviously, if you put hydrophones in for species that are known, to vocalize, I think you'd have relatively good year-round coverage. Outside of that, I think, you could imagine just increasing the frequency of sampling, maybe relying on some sort of study fleet utilizing commercial recreational fishers who are in the area to do some more kind of regular sampling at a reduced cost. But it's a difficult and expensive thing to do, especially given that a lot of these sites are in relatively remote areas on the shelf edge, and so they're not easily accessible. And the water depths are deep enough that kind of precludes the use of some methodologies, things like real-time camera systems and things like that, research buoys and the likes. So, yeah.
Okay. Thank you.
Yeah, I also wonder if something like eDNA could be useful for sampling some of these areas, trying to get down to depth and get some samples down there.
That's a great shout, Chip, actually. Certainly, I think for presence absence, eDNA it's pretty effective at answering those kinds of questions. I think with respect to looking at densities and abundance, my understanding at least is that the eDNA isn't quite there yet. But if we just wanted to look at presence of certain species in the area at certain times of year, then maybe eDNA would help us get at that too.
All right, there was a question written online. While the study confirmed that these closed sites do serve as spawning areas for multiple species, is there any evidence to support the assumption that these closed sites translate to more fish in the population than areas not closed to fishing?
The short answer, I guess, would be there's no evidence, but that's in part because of a lack of sampling in adjacent areas and control sites. The one comparison that we have made and that I feel comfortable making is looking at the age distribution of the individuals that we did collect and seeing how that compares to other fisheries independent sampling in the region. And I can say that with like take Georgetown Hole, for example, and scamp, one of the species that we did have decent sample size for, it's you know, a lot of the CMAP surveys indicate that scamp that are older than 8 or 9 years are in relatively low densities in the population, and the mean age of scamp that we collected at that site was a lot higher than I think indications from some of these fisheries independent surveys. But that's really like I say, one of the few, I think, kind of comparisons to areas outside the SMZs that I would feel comfortable making.
Go ahead, Brendan.
Just to jump in, I certainly agree with everything that Lewis just said. I think it's a valid question. To evaluate that question, unfortunately, it would take a lot more time and money. Certainly more time and money than we had before the sunset of the periods in question. But I will say that there are related closures in the South Atlantic, the deep water marine protected areas, and there are a couple of papers out there that did look at the closed areas versus open, I should say, adjacent control sites. And so if folks are interested, I can dig up those links and pop them in the chat. Maybe Chip can make them visible to everyone.
Sure. I can do that, or I can pass them along after the end. Please email me if you have some questions after this. I'll put my email address in the chat. Any other questions while I'm typing away? And I'll pass along those papers for you, Brendan. Yep. Sorry for the dead air. My brain only works in one thing at a time nowadays. Hopefully you can see all the information that Brendan had provided, a couple different papers. If you want to read some more on this topic that was brought up. Not seeing any other questions or hands raised. So let me start off by thanking the team for putting together a wonderful presentation and taking the time to answer the questions, and everyone else for attending the webinar today. I hope you found it very interesting. If you have any ideas for webinars in the future, please don't hesitate to email me, and I'll work on trying to get that integrated into our schedule. Before I give the authors or the presenters the last chance to speak, I do also want to recognize a couple of folks that are on the webinar right now today that were part of the steering committee setting up the sampling design for this. Mary Connolly was very important to setting it up. She's with the Nature Conservancy. Will Heyman, who was previously with LGL, has been actively engaged in this process, making sure that we collected samples in the spawning special management zones. And then, not without least, Nicole Pale. She has been instrumental in getting this working group together, keeping them on schedule, making sure that we're meeting our deadlines, and then Kara Chancellor for putting all this information together in a very elegant presentation. So with that, I want to thank the team, and then presenters, if you have any last comments.
Nothing for me. Thanks, Chip.
Thank you.
Yeah, nothing more to add, Chip. Thank you.
All right, everyone, have a good afternoon. David, I hope you're starting to feel better.
Thank you.
You too, Chip. Take care.
Take care.